Yeah, okay, so a short introduction. My name is Anastasia, and I will be giving you lectures in atomic force microscopy, and Eugene will be responsible for the practical course. I've graduated from Moscow State University, got my PhD in bioengineering in Hong Kong University of Science and Technology, and now I work as a senior research scientist for a company that does in vitro diagnostics and produces equipment for that and for hospitals. Thank you very much for joining this course. I hope that it will be interesting for you. And then if you don't have any questions from the organizational point of view, I will start the presentation. And and I will explain a bit more about the structure of the course and the grading system while I'll share my screen. So today there will be introduction to nanomechanics and general introduction in AFM instrumentation. So in this course, we will discuss with you working principles of AFM, some distance-dependent interactions. I think most of the time we will spend on different AFM modes because I mean, it is very important how exactly you analyze your sample and you get the images. Eugene will help you and teach you how to do sample preparation and some image processing and analysis and using the microscope that you have in the university. We also will discuss image artifacts and noise because it is not only only important to take the images, but it is also important to understand what exactly you see there, because there are a lot of artifacts can be acquired during the acquisition process. And last two lectures will be dedicated to your small projects. So I will ask you to prepare a short presentation about different applications, and all of us will discuss applications where you can use AFM, how it's being used nowadays, and so on. Today, we will have general introduction to AFM, and I will just show major principles of AFM. AFM operates on a very different principle than other forms of microscopy, such as optical microscopy or electron microscopy. The key component of an AFM is a cantilever, and it bends on response of to forces that it experiences as it touches another surface. Forces are actually very small, a few piconewtons, and can be detected and probed with AFM. And today, what I'd like to do is to start the course by having a general discussion of intramolecular and intermolecular interactions, and sort of like the stage when we can that's what we are going to cover like this first week. And first topic discussed is the forces that that result between the tip here and the substrate, and how we can calculate those forces. And so, okay, let's go to the next slide. So it's just a general reminder of some physics that you probably know already. So today I want to talk about electrostatic nature of the interactions, and they all can be traced back to the application of Coulomb's law. And Coulomb's law just says that if you have 2 point charges and those point charges are separated by a distance z, then there is an electrostatic force between the two charges, and that electrostatic force varies as 1 over 1 over the distance squared times of the product of these charges Q1 and Q2. These forces can be measured inside of a material or a medium which has a dielectric constant kappa. So if these two charges are embedded, let's say, in a liquid or in a solid, the interaction force that's experienced between the two charges is reduced by this parameter kappa. So that's also something that has to be taken into account when we discuss the interaction between point charges. So fundamentally, this is the classical picture of electrostatic interactions, and it's going to form the basis of basically our discussion throughout the beginning of this course. So I also like to emphasize another concept from introductory course of electricity. So it's electrostatics, very important. And you might recall that if you have two charges, one is fixed and called Q1, and another one is movable and called Q2, then there is a certain amount of work required to move the charge Q2 in the presence of the charge Q1. So here we start moving Q2 at some position far to the right, and then we want to relocate it to the left side, to some final position that we call distance Z. We have to integrate the components of work over the entire length of the path from some initial position, which is usually chosen to be infinity for some final position at the distance Z. So once that work is calculated, we can then formulate something called the electrostatic potential energy. That's how much energy this final charge distribution has assumed after it's been assembled, and that electrostatic interaction potential energy will be referred as a capital U. So again, it will be a function of the distance of separation at their final resting place. They're stuck down, so they're not free to move. I'm talking about the charges, right? Under some electrostatic forces, and they're exerted by these charges. So this is also that we just assume you understand, and the charges are basically just stuck down into the final position, and we are more interested in calculating how much energy is required to assemble the charges into a final orientation. So the interaction potential energy is useful because if you take the negative derivative of the interaction of potential energy, you'll get the force that acts on these charges. So for instance, in the case of 2-point charges, the interaction potential energy varies is like 1 over the separation between the two charges, and that depends on the product of two charges, Q1 and Q2. So Coulomb's law can be straightforwardly recovered from this expression by just taking negative derivative of it. Okay, and let's move to the next slide. So now it is important for you to remember that this quantity of energy U is a signed quantity. The value, like the polarity, the sign of U, whether it's positive or negative, depends on the polarity of the charges involved in our discussion. So here you can see it in this diagram. So we have our charge signed as Q, and let's say it is fixed here. And then we have two movable charges. They have opposite polarities, plus Q and minus Q. And that we would like to do is we would like to consider the motion of each charge in a turn, and then try to see how the electrostatic potential energy varies while we're moving these charges closer to the fixed charge, the capital Q. And again, it's just a reminder that if the polarity of two charges are the same, so that means that if capital Q and small q are both positive or both negative, the electrostatic potential energy increases as the separation between two charges. So, and then conversely, if polarity of the two charges is opposite, then interaction potential energy will decrease. So follow the blue curve indicated in this diagram. The interaction potential energy becomes negative. And that indicates that the final configuration of charges is in a lower potential energy state than it was in the beginning of the beginning of this calculation, when the two charges were far apart from each other. So nature always tends to go to the lowest energy state. And so given a choice, if the electrostatic potential energy is negative, that indicates that it's an attractive type of interaction. Then the charges in question can, well, they tend to go to the final position together. And also, I think it is important to discuss the difference between intramolecular interactions and intermolecular interactions. So, especially if we're talking about electrostatic potential energy. So to start this talk, let's focus on the intramolecular interactions. So these are the forces or the interaction potential energies that are appropriate for charges inside of a molecule. And to pick a molecule to start the discussion, let's pick the simplest one. It's just the hydrogen molecule, H2. The hydrogen atom, of course, is comprised of one electron and one proton to form a hydrogen atom. So hydrogen molecule is two hydrogen atoms pushed up one next to other by some distance. So if you just calculate the electrostatic potential energy interactions between the various parts of the atoms, you'll find out that there are four contributions that must be taken into account. First of all, this is a quantum mechanical calculation in principle, because the electrons basically are delocalized around the nucleus of each hydrogen atom. Those electrons have to be represented by wave functions, and those wave functions are schematically shown here by a spherically symmetric electron charge cloud. What also is happening is the two atoms are brought close to one to another, and the nuclear, they are like positive nucleus of the two atoms can repel each other. Then this is a repulsive interaction, and that will be taken into account as well. Likewise, two electrons associated with each hydrogen atom. Those electrons have negative charges. So these charges will repel, and they will introduce a repulsive interaction as two atoms are pushed closer one to another. To counteract these all repulsive interactions, they're going to be, there should be an attractive interaction between the electron in the atom on the left and then the nucleus of the atom on the right. That will be an attractive interaction, and there will be the same thing from the right atom and the right electron and the left nuclear. So they calculate whether the hydrogen molecule has a stable separation between the two hydrogen atoms. You have to calculate electrostatic interaction potential energies, including everything that I just was talking about. You have to sum them up, and if there is a minimum in the electrostatic potential energy, then the location of that minimum tells you where one hydrogen atom is going to be located with respect to the other. So that whole process is again schematically indicated by this graph on the bottom, so the dashed curve, the black one, shows the typical electrostatic interaction potential energy between these two hydrogen atoms as they brought close one to another. As you can see, there will be well-defined minimum. It's about minus 4.5 electron volts, and it occurs when the two atoms are about 75 picometers apart. So this defines the equilibrium separation between the two hydrogen atoms, and then leads to the stable hydrogen molecule that we can observe. So this whole process is referred as a chemical bonding in a generic way. Okay, so we saw that some molecules have some molecules, how they work, electrically neutral molecules can develop a dipole moment simply because different atoms attract electrons within the molecule in different ways. And this very subtle redistribution of charges within a molecule gives the rise of dipole moment. And what we're going to discuss now is how these dipole moments of different molecules can interact one with another and produce some certain effects. So let's review the definition of a dipole moment. You have two charges, plus q and minus q. They're separated by distance d. Then we say a dipole moment. Here I showed as a symbol p. Develops. So this this dipole moment develops. It has a direction, so it's a vector, and there is a vector sign over it, you can see here. And the magnitude of this dipole moment is just the product of the charge that has been separated times the distance d over which separation occurs. So the magnitude of the dipole moment p is just an equal product of q times d. So we need a system of units to measure dipole moments, and the system of units historically has been referred as dBi. So dBi unit is a bit old measure. Basically, a molecule has a dipole moment of 1 dBi, means that there is about 2/10 of electrons separated one from another by about 1/10 of nanometer. Yes, so 1 dBi is about 3.3.10 power minus 30 coulomb meters, and this is another unit of dipole moment that can be used as well. Okay, so now let's talk about dipolar covalent bonds and ionic bonds. So this again, just a reminder. So again, electrically neutral molecules can have a dipole moment, and interactions between molecules is not equal to 0. And so depending on the polarity of the atoms, the bonds, the chemical bonds can be varied from not polar covalent bond to polar covalent bond and to ionic bond. Depending on this how big is the charge, then we have different forces between two atoms. So now let's talk about intermolecular interactions. So all intermolecular forces act as a charge species. the intramolecular interactions all occur because molecules or atoms interact one with another with a wide variety of different electrostatic interactions. You can have the column interactions between point charges. This would correspond to ion-ion interactions. You can have atoms or molecules that have permanent dipole associated with them. These will give you this will give you a dipole-dipole interactions, and they will give you an electric field for one molecule. This will be referred as an induced dipole moment. And the incidence dipole can be also associated with rapid electron movement, as one molecule becomes correlated with rapid electron movement in another molecule, and this will be called dispersion intermolecular interaction. Okay, so now I want to talk about quite important interactions in as a van der Waals bonded. So van der Waals bond actually is a sum of three long-range attractive forces. As a key-sum force, Debye force, and London force. So key-sum interactions are the electric interactions, electrostatic interactions among the permanent dipoles of polar molecules here, which mutually modify their orientations to align their dipoles. So they arise when we have plus at the end of one dipole molecule is attracted to the minus at the end of another dipole molecule. And this is the full calculations for key-sum force. So we talked a bit about Debye force already, is here. And then London force. London is dispersion interaction, and they arise when a temporary imbalance of electrons of a non-polar molecule induce a temporary dipole in another nearby molecule. So the sum of these three interaction potential energies will compose the van der Waals force. The important thing is to recognize that each interaction potential energy varies as 1 by z to the 6th power of the separation between the two molecules that are interacting, and the characteristic features of each term that you should be able to recognize quite easily. So it's for key-sum force, for Debye force, and for London force everywhere. So van der Waals forces, you will see them in other discussions when we will be talking about different AFM modes. So I don't want you to remember all these terms here. What I want you to remember is this z power 6 over here. It is quite important. Okay, so the combination of these interactions that we discussed result in a force distance curve similar to what you can see here. So as the tip is brought towards the sample, van der Waals force causes attraction. As the tip goes closer to the sample, obviously this attraction increases. However, at small separation, the repulsive coulombic force becomes dominant, and the repulsive force causes the cantilever to bend as the tip is brought closer to the surface. So there are other interactions beside coulombic and van der Waals forces which can have effect. So for example, when AFM is performed in ambient air, the sample and tip might be coated with a thin layer of fluid, for example, water. Then the tip, when tip comes closer to the surface, then capillary forces can arise between the tip and surface. So if you need to calculate forces, you should not forget about these details as well. Okay, so that was like a general introduction into different forces. So let's talk more about AFM. So why AFM? I want to start talking first from the optical microscopy. So when light from the various points of specimen passes through the objective, right? It is reconstituted as an image, and the various points of our sample appear in the image as a small patterns. And if 2 points are too close to each other, so these patterns are called Airy Disks. So this Airy disk can merge together, basically. And this phenomenon is caused by diffraction or scattering of the light as it passes through the parts of the sample. And another thing that also causes this is a circular rare aperture of the objective in the optical microscope. So the limit up to which 2 small objects can be still seen as a separate entities is used as a measure of the resolving power of an optical microscope. So the distance where the limit is reached the limit is reached is known as effective resolution of the microscope, and usually it is denoted as D. The resolution. So this resolution is a value that can be derived theoretically, giving the different optical parameters of the instrument and also the average wavelength of illumination. So it is also important to know that the objective and the tube lens do not image a point in the object. So they usually image these airy disks with diffraction rings. And an airy disk is the region enclosed by the first minimum of the the first minimum of the Airy pattern, and contains approximately 84% of the all luminous energy, as you can see here. And then everything else is shown as a small peaks here. So the resolution between 2 points, let's say X and Y, depends on the length of the wavelength of the light, and divided by refractive index of the imaging medium, and combined with sinus alpha. So this combination, nu and sinus alpha, is known as the objective numerical aperture. These values you can usually see on the objectives when if you work with a microscope. And it's a very important value. It shows you the resolution, basically. Objectives commonly used in the microscopy have this numerical aperture that is less than 1.5 as a value. This means that it's about, so these alpha, this is the degrees, it's about 70 degrees here. And theoretical resolution limit is the shortest practical wavelength, and it is known to be approximately 400 nanometers. So, this is basically the limit of the optical microscopy. And what is these 150 nanometers? So Z for 400 nanometers is the shortest practical wavelength, and 150 nanometers is the lateral dimension when this wavelength is approaching our sample. So it is quite hard to reach this resolution limit, so that's why we have not only optical microscopy but other microscopies as well. So another type of microscopy that you've probably known is electron microscopy. So this is a schematics of electron microscope, and the electronic beam is shown here as a dashed line. Some types of transmission electron microscopes have atomic resolution, and it is very handy to use them. The problem is that they're quite expensive, and sample preparation is very laborious, because samples, first of all, they must be electron transparent. This means that the thickness of the sample that you can use is very, very, very thin. So so you cannot image like different like tissues, or you have to do the special sample preparation for that. Second, in order to get the fine beam, we use vacuum chamber. So the sample must tolerate the vacuum chamber, so it cannot be anything live. And again, it should be small enough to fit the chamber. So there's a huge microscope, but the chamber for samples is quite small. And transmission electron microscopes, they're widely used in science, but for biological applications, we usually use it for dry samples, or sometimes we have cryo transmission electron microscopy when we can visualize different proteins, and hoping that these proteins still have their natural state. So in biology, the application is limited by these requirements for the sample. That's why we decided to introduce this course with AFM, and the grandfather of AFM is scanning tunneling microscope, or STM. So the scanning tunneling microscope works by scanning a very like a very sharp metal wire tip over a surface. So it's bringing the tip very close, and it applies vertical. It applies electrical voltage to the tip of the sample. So that's how we can image the surface at an extremely small scale. We can also see individual atoms here. So first, it was developed by Gerd Binning and Henrik Roher in IBM lab, and after several years they got a Nobel Prize in physics by developing this microscope. So it has three working principles. Tunneling, I will talk about this in the next slide. It has piezoelectric effect that it uses to acquire the image, and it has very important feedback loop to adjust the height of the metal of this tip. Okay, so what is tunneling? Tunneling is a quantum mechanical effect. A tunneling current occurs when electrons move through a barrier that they classically shouldn't be able to move through. In classical terms, if you don't have enough energy to move over the barrier, you won't move. However, in the quantum mechanical world, electrons have wave-like properties. So these waves don't end abruptly at wall or a barrier, but it kind of tapers off quickly. If the barrier is thin enough, the probability function may extend into the next region through the barrier. And because of the small probability of an electron being on another side of the barrier, giving enough electrons, some will indeed move through this barrier and appear on another side. So when electrons move through the barrier in this fashion, it is called tunneling. So here you can see the voltage of the potential barrier and how electron moves via this barrier. The scanning microscope basically is the predecessor of the atomic force microscopy, a near field optical microscope. And so one of these main principles is using tunneling current. For tunneling current, we have basically two requirements. First is a small distance, because we need to overlap electron wave functions. And second is a bias for net current flow. In a metal, the energy levels of the electrons are filled up with a particular energy known as a Fermi energy. And in order for an electron to leave the metal, it needs an additional amount of energy. It's called a work function. And then when the specimen and the tip are brought close to each other, close enough, there is only a narrow region of empty space that is left between them. So the tip does not actually touch the surface of the sample. On both sides, the electrons are present up to this Fermi energy, and they needed to overcome a barrier of this empty space to travel from tip to specimen and vice versa. So if this distance is small enough, electrons can tunnel through the vacuum barrier. And then when a voltage V is applied between specimen and tip, the tunneling effect results in a net electron current. And this is what we can actually detect. Okay. So this is about the feedback loop. So this is a schematics of feedback control when the force sensor senses a charge in a sample, the change in a sample height and the piezoelectric moves to maintain the sample tip to surface distance. So here we have piezo element and force sensor. This is the tip, this is the surface. So so the elements are moving along the surface, and if decreasing force is detected, this means there is a bump here going down, then piezoelectric element moves the tip down. If the increasing force detected, then piezoelectric element moves the tip up, and then that's how we maintain the same distance between the tip and the sample, and this is called feedback loop control. As I've already mentioned, there's a force sensor. So the force between AFM probe and the surface is measured by this force sensor transducer. As it illustrated here, the probe comes into contact with the surface, the voltage output from the transducer increases. It is important that the output of the transducer is monotonic and increases as greater force is applied between the probe and surface. Force transducers may be constructed that they measure forces as low as 10 piconewtons between a probe and a surface. And typically, the force transducer in AFM is a cantilever with integrated tip, as we call it, the probe, and the optical lever. However, there might be different types of force sensors that can be used in AFM. In a force transducer, it is used to measure this interactive force between the tip and sample surface. That's how feedback loop works. And now we will talk about piezoelectric effect. This effect was first discovered by Pierre Curie in about 1880s, and STM use inverse piezoelectric effect. So by applying voltage on the material will deform. So here was some sort of material, we apply some voltage, and then it will deform. That's the piezoelectric effect. So the piezoelectric materials used in STM are various of kinds of different, like lead zirconate or lead titanate ceramics, and so there are different modifications of these materials. Piezoelectric materials, they will convert electrical potential into mechanical motion. And they are naturally occurring like as a crystalline, amorphous, or polymeric materials. And in AFM, we generally use synthetic ceramic materials, as I already mentioned. These piezoelectric elements can react in a very precise way. So some of them, they can change their shape in 0.1 nanometer when we apply some voltage. So this is very precise. And in STM, there are different types of piezoelectric elements can be used. Originally, they used a tripod scanner as depicted here. So it has 3 axes here, and then that's how the element was navigating along the surface here. So this is piezo element depicted here is the same thing as this tripod scanner or tube scanner. So nowadays, tube scanner is mostly used piezoelectric element. How it works, it has certain it's like basically a tube with certain thickness, and when we apply voltage, then this thickness is working to deform these tube. And probably you have a question, what is STM and what is AFM, how similar or different they are. AFM actually caused a revolution after STM was developed because it is very cheap and it is extremely high resolution microscope. Both AFM and STM are referred as scanning probe microscopy, or SPM. So please do not mix tunneling microscope, STM, and probe microscopy, SPM. So again, STM uses tunneling current between the metallic tip, and it is conducted, and it has to use conducting substrate because we're using tunneling effect. And there is no physical contact between the probe and the tip, and the sample. But AFM uses van der Waals force between the tip and surface, and it actually touches the surface of the sample. I think here is the schematics of STM and AFM, and please do not forget that there is always a certain distance between the tip and the sample in STM. And this picture shows basically the similarities and differences between AFM and STM again. So AFM employs an optical beam bounce based feedback, which consists of mechanically sharp tip attached to a cantilever. And when tip comes to a contact with the sample, it starts to scan. So these forces, again, can be attractive or repulsive in nature. And overall force curve between the tip and sample is shown in this figure. So the attractive region is due to van der Waals forces, and the region where scanning is done is not in non-contact mode. However, the curve for STM is a bit different, again because of this distance between the tip and the sample. If we compare AFM with traditional microscopes, let's say optical microscope or scanning electron microscope, and what kind of dimensions we can measure. So in practice, the measurements the areas greater than 100 micrometers, it is hard to measure with AFM and doesn't make much sense. But for higher resolution, goes to like atomic resolution, AFM is very good example of the application of the microscopy. And at the end of this lecture, we'll talk a bit about key inventions and applications of AFM. So you can use these paper as the starting paper for your research in AFM applications, because it has a lot of examples that you can use in your presentations. So this is the timeline, right? And key inventions developed over the years include an optical system and fluid cell enabling contact mode AFM to operate in aqueous solution called bio-AFM, a dynamic mode AFM, which oscillates the AFM tip to reduce friction while contouring the biological sample. Then force distance curve based AFM. So it contours the surface of a biological system while recording pixel by pixel a full force distance curve, and multiple parametric AFM here. So multiparametric AFM contours the sample while mapping multiple physics or chemical properties. Here, MR-AFM is molecular recognition AFM. It images and maps specific interactions of biological samples. MF-AFM is multi-frequency AFM. It contours the sample while oscillating the cantilever tip at multiple frequencies. And that's how it maps various physical parameters. And it's quite similar to optical imaging AFM or opto-AFM for imaging complex biological systems. And the one of the recent developments is high-speed AFM. So it speeds up the image acquisition time by a factor of 1,000 and provides access to dynamic processes in biology. Most modes actually, how to say, it's not like bio-AFM works only with certain samples. The dynamic mode AFM works with certain samples. So these types of AFM, they kind of overlapping with each other. So please don't be confused when in some papers they use different parameters to measure the same thing. And because during this course, we will be mostly focusing on the biological systems. So I will just explain a bit more about different modes of AFM in application to biology. The key breakthrough that led biological AFM was the development of an optical detection system, followed by the design of a fluid chamber enabling imaging in buffer solution. So this means that we can maintain the native state of the biological system. So what does it mean? This means that the cells can be alive. We can measure them in the native state. So here's the example of measuring of the transmembrane proteins here while the cell is still alive. So this is a great advantage over, for example, a transmission electron microscope that I was talking about here, because here we can measure transmembrane, make an image of a transmembrane protein, but we cannot guarantee that it will be in its native state because we use vacuum, and I'm not sure how strong is your biological background. So for transmembrane proteins, it is very important in what environment they're presenting. So for example, in this transmembrane region, they have more hydrophobic amino acids rather than on the surface. Let's say outside or inside of the membrane, and it is very important to keep the membrane around them if we want to see them in their native state. So this is one of the problems that people face when they try to crystallize this kind of proteins, because if you try to crystallize it without membrane, then it will not be in its native state, and it is not relevant for the further research. Okay. So next, what I want to talk about is that when we go from the FD AFM, FD is a first distant curve based AFM, to different multi-parameter imaging. The question came up that whether AFM can do more than just contouring a surface, because probably you've already understood that we cannot probe anything what is inside the sample. We just go along the surface, and what we record is basically different bumps. So for example, here, what you can see is DNA. So this image just shows the difference of the height between the substrate and this DNA, but it does not tell it doesn't tell us what is the chemical composition of DNA, what nucleotides are here, and so on. So the main milestone was the realization that simultaneously with structural imaging, AFM is able to provide some biophysical properties. So initially, such properties were measured by approaching the AFM tip to the surface and then retracting it from the biological sample while recording this single force distance curve. That's why this AFM is called force distance curve-based AFM. So approach when this force distance curve can quantify the height, the surface forces, and mechanical deformation of the sample. Also, it can give us the values of the elastic modulus and energy dissipation. And retraction force distance curves allow adhesion forces to be measured. So we'll talk about adhesion forces next at the next lecture. And currently, the most widely used application of force distance based AFM is the mapping of the mechanical properties of biological systems. This is quite important because, for example, different cellular functions rely on mechanical properties. So now I have a question to you. Can you please give me any examples of measuring adhesion forces or elastic modulus in cells? Why we need that? Why we need this? Can you think about what kind of molecules or what kind of processes we can measure using these kind of AFM. And so the question is where we can use this information about adhesion forces and elastic modulus in a live cell. So what kind of structures there might be interesting for us to measure this? Cytomembrane. Okay. Any other ideas? Receptors. What kind of receptors? Because usually receptors on this cell surface, they are more chemical based, so they detect some small molecules or big proteins. But here what we're interested in is the physical parameters. So basically how much this thing can bend. That's what elastic modulus is. Okay, so thank you, Yi-Chan and Bella, for your suggestions. Yeah, I will just talk a bit more. So these curves show us the physical properties of the cell. So one of the objects that we are really interested in can be cytoskeleton, because if you remember how, for example, a fibroblast looks like, or different immune cells that go into tissues and then kind of go through around other cells. So they change their shape using their cytoskeleton. And the work of the cytoskeleton induces the deformation of the cell membrane and changing a bit of its properties. So what we can use, we can check how this cytoskeleton can be changed if, let's say, we apply different drugs to this cell, or we have any treatment of this cell. And another example, we can spatially map the stiffness of, for example, actomyosin cortex, because actomyosin, it's not only the actin and myosin that is not only present in our muscles, but also present in cultured cells. And there are two different types of cell cultures, the suspended cells and the adherent cells. And then sometimes when we treat cells with different drugs, the force with what they adherent, they can go away from the surface, and we sometimes can assess how strongly they are attached to the surface or not attached to the surface. Another example is, for example, is mapping yeast cells. So when they divide, they form buds, and then like two cells separate, and then on both of these cells they have a scar from this budding, and we can also check how stiffness of this scar on the surface of the yeast cell is different from the rest of the membrane. So one of the issues of, for example, this kind of AFM, force-based AFM, is the temporal resolution and lateral resolution, because we can measure the elastic modulus, for example, and if the cell is moving at this particular moment, then what we can experience is the tip to sample drift. And then when the drift occurs, then the resolution falls, like worsens dramatically. And this is one of the difficulties with like working when we apply FM measurements to live cells in different solutions, for example. Okay, then next one that I want to talk about a bit in more details is multi-frequency imaging. So multi-frequency imaging, or MF-AFM, it can map, again, mechanical and functional properties of the biological sample. But the good thing, especially in comparison with force-based AFM. Force-based AFM is quite slow, and not slow, it kind of gives us too much of data and it requires a lot of acquisition time. So multi-frequency AFM is one of the advanced dynamic mode AFM, and it can work with high frame rates. Also, it involves different frequencies of cantilever motion as well. So it can be very useful for example, imaging of water layers covering different proteins with very, very, very small forces. And it will help us to understand how these proteins are changing their shape, and then how they work with very high precision. Yes, and then the last mode that I want to talk about with more details is high-speed AFM, because it was recently a huge, how to say, very popular method. And if we compare it with, let's say, fluorescent microscopy, in general, AFM imaging is very limited in terms of how quickly we can acquire the changes. So in comparison with fluorescent microscopy, because fluorescent labels and dyes, they can blink very quickly and we can detect the information with high time frames. But high-speed AFM was developed to kind of breach this kind of gap between fluorescent microscopy and AFM microscopy. And again, coming back to the way how AFM microscope is assembled, the slowest part in AFM is the cantilever, because everything else is the acquisition of the forces here is very quick process, but the movement of the cantilever, deforming the cantilever and so on, is quite slow process. So the high-speed AFM tries to overcome all these drawbacks. And so first of all, the cantilevers that are used in high-speed AFM, they have a bit different spring constant, and that's how this gap is kind of narrowed. And in the very, very early days, high-speed AFM, they were used only for checking the changes of the DNA 3D structure, and work of myosin, different chaperone systems, and so on. In recent days, AFM with high-speed properties can allow us to understand how the motor of ATPase work. So ATPase, just to remind you, is one of the main proteins in mitochondria, and it looks like a motor, like spins around, and it has different subunits, and they rotate as a water mill basically. And this rotation is very hard to detect. And this rotation is working by 3 chemical states: when the protein is empty, when it binds ATP, and when ATP decays to ADP, D-phosphate, and inorganic phosphate. So high-speed AFM allows us to image this kind of motion, and it is quite cool, I would say, and it's one of the main applications of high-speed AFM. So this is the general comparison of all high-resolution techniques in molecular and cell biology. So again, atomic force microscopy that we are interested in, the resolution is less than 1 nanometers, but again, to achieve atomic resolution in this microscopy, we require certain level of sample preparation. So the AFM supports samples in different conditions. So you can measure the samples in air, in liquid, with different temperatures, and so on. One of the main artifacts, so some images that are not produced by the changes in the sample, they come from wrong tip type, or like broken tip, or so on. A different force, sometimes you use too much of force or too small force, and also the scanning rate. We'll discuss with you a bit later. Depends on the scanning speed. You can get some strange results on the AFM. As an advantage, we can image under native conditions with very, very, very high resolution. We do not need any staining. So those of you who worked with fluorescent microscopy, confocal microscopy, and so on probably know how much time you spend for fluorescent imaging, staining, and how many artifacts it can produce. One of the things that we still need, sometimes we need to label or fix our sample. Again, just because we are working with very high resolution and we cannot afford sample moving around too much. Another good thing is high signal to noise ratio. And in parallel, you can assess physical properties and biological properties at the same time. Limitation is that, again, we basically measuring the surface. We cannot tell what is the sample made from, and so on. Another super resolution microscopy that will not be covered in this course, they have a bit worse resolution and require fluorescent labeling for imaging. And one of the drawbacks of any fluorescent labeling is the bleaching of the dyes, because you cannot excite them for too long, otherwise they just like lose their properties. And a good thing that you can see the three-dimensional cellular structures, and it's a bit easy to monitor biological process in live cells when you use super resolution microscopy. But again, the sample preparation here is very laborious. So all this super resolution microscopy, what they do, they use blinking of fluorescent labels, and they take huge amount of pictures, and then they kind of superimpose all these pictures together, and then that's how you get the image. So next one is transmission electron microscopy. It's about 10 nanometer resolution. Sample preparation should be, so it requires dehydration, special type of staining, and it should be very, very small and thin sample. As an artifact, all crystals are deformation of the membranes and so on. You can see there. And again, you cannot take pictures of the same place for a long time, because you can just burn a hole in your sample, so-called beam damage. As advantages, it solves atomic structures of proteins, especially for cryo-electron microscopy. Cryo-electron microscopy is also used for actually understanding the protein structure, and a lot of models in PDB bank actually made from using cryo-electron microscopy. But one of the huge limitation, no live processes. So all the samples are fixed and dead. And the last one is scanning electron microscopy. The same, almost the same resolution as transmission electron microscopy. One of the things is that you have to dry the sample out. You cannot measure anything in the solution in scanning electron microscopy in comparison with atomic force microscopy. So the images, I would say, they might be similar. So if you take an image, for example, from gold nanoparticles, you can take an image using atomic force microscopy and scanning electron microscopy, and the quality of the images can be quite similar. But again, you cannot measure these nanoparticles in the solution, for example. And yes, it is highly used to image surfaces of tissues and cells, but most of the application of scanning electron microscopy is for nanoscience, like nanoparticles, nanotubes, and so on, because it does not allow any live processes to be imaged, because everything should be dried. Okay, so I think this is all, basically this is all for today. To read, you can review all the applications in the paper that I was mentioning, and general introduction to scanning probe microscopy you can also find here. We also added textbook to Moodle. Please check it. So thank you everyone, and I think this is all for today.
С вами был Тибериус. Если видео понравилось, ставьте лайк. Если видео не понравилось, так уж и быть, ставьте дизлайк. Но обязательно напишите почему. Подписывайтесь на канал. Всем пока. До скорого.